Hi, my name is Xavier Lapointe. I'm a master's student at Sherbrooke University in Marie Brunet's lab, and today I'll be introducing a new deep learning model that we built and trained to perform segmentation of coding regions within transcripts. In the past couple of years, new technologies such as RiboSeq have allowed to expand the landscape of the human proteome. And for the first time last year, major actors of genome annotation such as GenCode came together to build a catalog of over 7,000 non-canonical open reading frames supported by RiboSeq evidence. Some of these ORFs are located upstream, downstream, or can overlap with a canonical CDS, while others are annotated on transcripts previously thought of as non-coding. And while the recognition of these ORFs as active members of the translatome is a big step towards an exhaustive annotation of the human proteome, one class of open reading frames that remains excluded from functional annotations are those within pseudogenes. As a reminder, pseudogenes are copies of other genes which have presumably lost their original function, either because of mutations to the CDS or because of a lack of regulatory elements essential for gene expression. There's two main classes, those obtained by duplication and those obtained by retrotransposition of a parental gene's mRNA. Pseudogenes are annotated and considered as non-functional, therefore no CDS is annotated within pseudogenes, and they are often excluded from functional omics analysis, mainly to avoid introducing any ambiguities since they're very similar to their parental gene. However, thousands of pseudogenes are transcribed, and many of the best detected non-canonical ORFs are encoded in pseudogenes when we look in proteogenomics database such as OpenProt. And our goal with this project is to use deep learning to provide an unbiased assessment of the functional orpheome, inclusive towards any class of non-canonical ORFs. Most deep learning approaches aiming to predict translation events rely on the use of a sliding window, extracting local features relevant to translation initiation site prediction. The downside of this approach is that these models cannot leverage long-range information since they're not provided the full context, which is the transcript. They're also very computationally and time expensive. To address these problems, we suggest an end-to-end approach where we simply feed a full one-hot encoded transcript to the model, and the model outputs predictions for every position of the input sequence. Then, of course, a loss is calculated by measuring the difference between the output and the target vector, where we label every nucleotide of a coding sequence with 1s and the remaining with 0s. Our model is named FOMONet, which stands for Fear of Missing Open Reading Frames Neural Network. The FOMONet architecture is derived from the UNet, which has been extensively used in computer vision and image segmentation. And like the UNet, FOMONet is based on a encoder-decoder architecture, where the encoder compresses the input sequence into lower dimensional representations while increasing the number of feature maps. The decoder does the exact opposite to recover the original input dimensions. At the very end, we have a one-by-one convolution with a sigmoid output function to obtain an output sequence corresponding to coding probabilities for every position of the input sequence. Also notice the skip connections where we concatenate feature maps from the encoder to the corresponding decoder layer, and that allows to minimize the loss of information in the expanding path of the neural network. FOMONet differs from the UNet in a couple of ways. First, the nature of the input, which is one-dimensional instead of 2D. Also, the use of depthwise separable convolutions, which allow to reduce the number of parameters compared to a network based on conventional convolutions. And finally, we've added residual connections, which allows the gradient to flow more easily during backpropagation. Our dataset is made of about 16,000 protein coding transcripts from Ensemble 95, All of these transcripts have a transcript support level of 1. And we have randomly selected one transcript per gene and annotated the canonical coding sequence as the target. Then we've grouped transcripts coming from paralog genes and split those groups into 10 sets to perform cross-validation. We've evaluated FOMONET on the test sets of the 10 splits, and the generated ROC and PR curves corresponds to the mean curves of these 10 runs. As you can see, FOMONET grades very well in all the metrics for the task of predicting canonical human CDS. And to our knowledge, the only other end-to-end approach taking as input a full transcript is this transformer, which FOMONET outperforms in both ROC AUC and PR AUC metrics. So these are graphical representations of FOMONET output on two transcripts. The highlighted regions corresponds to the CDS, and these graphs display the output, which is the coding probability, on the y-axis for every nucleotide of a transcript. And as you can see, the output fits the edges of the region highlighted in red in both cases, even when the transcript coverage of the CDS is very low, like in the second case. So seeing these great results, we wondered whether the features of translation learned in humans could transpose to other eukaryotes. We took the FOMONET model trained in human and tested it on other species. To avoid any bias caused by sequence homology, we removed all transcripts coming from orthologous genes relative to humans. And interestingly, we observed very little decrease in performance, even in some distantly related eukaryotes such as C. elegans. Finally, as I mentioned earlier, one blind spot in current annotations is CDS prediction within pseudogenes. Thus, we tested FOMONET on a curated dataset of pseudogenes. We filtered the proteogenomic resource OpenProt to retrieve pseudogenes from which a CDS is supported by strong experimental evidence. This resulted in a bit under 1,800 transcripts, and FOMONET predicts a CDS with a very high confidence in about 700 of these cases, which bodes well for its ability to distinguish functional pseudogenes from disabled genomic copies. In summary, we have built a convolutional neural network which performs segmentation of transcripts to identify coding sequences. FOMONET outperforms state-of-the-art algorithms to predict canonical CDS in humans, and also performs on other eukaryotes with minimal decrease in performance. FOMONET is also able to predict non-canonical CDS within pseudogenes, filling this blind spot of current methodologies. Thank you for your attention. I'd like to thank YSCB and Great Lakes Bio for their generous travel award. I would also like to thank CIHR for my master's scholarship, and I would like to thank my PI Marie Brunet and other mentors from Sherbrooke University, Michel Scott, Pierre-Marc Jourdouin, and Pierre-Étienne Jacques. Thank you very much, and see you in Montreal.